Welcome back, everyone. My name is Dr. Jim Scully, and we're here for the fourth session focusing on kidney physiology, and we're going to talk about sodium today. This gives the outline on sodium. We'll start off with a few general comments. We'll move on to how indeed you and I sense a sodium deficit, and then we'll look at some important outcomes or responses to a sodium deficit. And then we'll finish off talking about a sodium excess. Not as common an issue that the physiology was designed to address, but I think by focusing on both a deficit and an excess, we can recapitulate some of the physiologic changes that occur in response to these changes. So here we are again. We've started off, remember in the first lecture, spending all of our time thinking about how we look at body fluid compartments. And then, of course, I inverted that beaker-like example to focus on the intracellular space, the interstitial space, and then the plasma volume up here at the top. And then we said that one of the first functions of the kidney related to accessing that plasma flow, and that the critical consequence of that was that blood flow from the plasma compartment went to the kidney glomerulus, where on the basis of Starling forces, the kidney creates an ultrafiltrate that enters the tubular lumen. When that enters the tubular lumen, we talked about the passage of fluid down through the different nephron segments of the kidney. So we focused on body fluid compartments, glomerular filtration rate, and tubular function. And when we talked about tubular function, we talked about the key ways in which the kidney utilizes sodium gradients to reabsorb sodium from the lumen, where it is presented into the kidney because of Starling forces, and how water follows that. So today we're going to talk about sodium. Now, one of the things that's important to recognize is that we have to have an intake, and a typical North American diet is about 150 milliequivalents of sodium a day. And indeed, you and I have to have a mechanism in place that allows us to balance intake and output. And I'm not going to focus too much on extrarenal sources of sodium loss, but really look at the kidney as if it's the final arbiter of sodium balance. So remember, what happens to that filtrate? Well, the first thing is that it filters an extraordinary volume of water. So on the basis of Starling forces, the glomerulus filters about 150 to 170 liters a day, many-fold greater than our total body water, and of course reabsorbs that. It also filters approximately 22,500 millimoles of sodium, much more than the total sodium content, and reabsorbs most of that. And so one of the things to recognize is that the set point, if you like, for physiological balance is that the kidney reabsorbs 99% or more of the water that is filtered and 99% or more of the sodium. So before we launch into how we sense sodium balance and how our kidneys are able to respond to signals and changes in sodium balance, let's look at one case here. And this is drawn from one of my partners many years ago. And this is a case of cholera in Haiti. Sadly, of course, we still are challenged today with cholera in Haiti. Let's imagine that you volunteer to work in a medical clinic and you see a 16-year-old girl who's brought in by her family. She started to have fever, abdominal cramps, and she developed a profuse watery diarrhea. She was losing as much as 1 liter an hour from her gastrointestinal tract. When you saw her, her blood pressure was 90/50. A normal value would be 130/80. Her heart rate was 130 beats per minute. Normal heart rate in a typical resting individual would be 70 to 80 beats per minute. She was very lethargic, unable to drink. Her eyes were sunken. She had poor tissue turgor. When we refer to tissue turgor, we talk about when we take a pinch of our skin. And then, of course, most of us, that skin will collapse again. Decreased tissue turgor means when you raise the skin like that, it tends to retain that distortion in its normal anatomy. So the question becomes, what are you going to do for her? And the thing to recognize is that the GI loss, the diarrhea that is a consequence of cholera in this setting, is a fluid that is very high in sodium. So what we're talking about is a clinical situation in which a patient develops a marked depletion in their total body sodium, and along with that, their total body water. So let's start off with some general comments about sodium. First, of course, it's critical for life. We actually began to get a sense of that during our very first lecture when we looked at body fluid compartments and the critical role that the movement of sodium in its retention in the extracellular space plays in terms of cellular physiology. And then we saw how, in fact, that low sodium was critical for the kidney's ability to manage salt and water as that glomerular filtrate transits the nephron. Interestingly, of course, the total sodium content has a major impact on the blood or plasma volume. And the plasma volume has a major impact on blood pressure, as you learned in your cardiovascular physiology section. And we know that blood pressure is a key determinant of tissue perfusion. So that sodium balance regulates, of course, the volume in the plasma compartment, which regulates blood pressure, and which is a key determinant of organ perfusion. Now, early humans, as we were evolving, running around the plains of East Africa, as you can imagine, evolved in a very low sodium environment. Salt was scarce. So one of the things that I like to think about is that, in fact, human kidneys evolved and were designed to retain sodium in order to maintain blood volume. The fact that the kidneys filter 22,500 milliequivalents of sodium a day is a challenge for them, because indeed they must reabsorb 99% or more of that sodium. If they excreted that sodium, then we would have, and as we evolved, our forebears would have been forced to seek sodium, and sodium was scarce in the environment. Now, of course, we sit in a somewhat different, certainly live in different times, and we're challenged in different ways today. In modern times, we likely eat too much sodium. And if we eat too much sodium, it turns out to be a not unimportant determinant of high blood pressure, which is one of the scourges of modern day life. Now, normal and recommended salt intake. So, for example, the minimum required for life. This is a somewhat arbitrary number, but it indicates the ability of the kidney to take that 22,500 milliequivalents filtered into Bowman space that transits the nephron to reabsorb all but about 10 to 20 millimoles, about 200 to 450 milligrams a day. In North America, and we're going to work with this number, the average sodium intake is about 150 millimoles per day. And the maximum, recommended maximum, is closer to, actually for health, is about 100 millimoles per day. And we in clinical medicine often try to restrict sodium intake, particularly in people who have heart failure and people who have high blood pressure, to between 70 and 100 millimoles per day. So 150 millimoles per day is more than we require to maintain daily health, but it is the amount that the kidney will have to excrete in order to avoid loss of sodium in the urine and gain in sodium in the total body water. So, how do you lose sodium? Well, you can lose sodium sweating. The average sodium concentration in sweat is about 35 millimoles per liter. So you can imagine that if you're out in a gymnasium working and sweating, or playing football, or wearing a heavy uniform on a hockey arena, you're going to lose salt. And we're not going to talk too much about the regulation of that. Diarrhea is an important, so gastrointestinal contents and diarrhea can be associated with significant losses of sodium, 100 to 120 millimoles per liter. And you can imagine this young girl sitting there in Haiti with cholera was losing about a liter of fluid from her gastrointestinal tract every hour, developing a considerable deficit in her total body sodium because of the content of sodium in the diarrheal fluid. Vomiting also causes a more modest loss of sodium, about 10 millimoles per liter. And osmotic diuresis, one of the common ways in which diabetes impacts on patients and accounts for part of the clinical presentation, is that if someone cannot control their glucose concentrations. As we talked about last lecture with glucose handling by the kidney, that glucose will end up in the lumen of the nephron, and the nephron will not be able to reabsorb it all. As it stays there, it will stay there with water. Urine flow rates within the tubule will increase, and that will cause sodium loss. And that's variable depending upon the level of blood glucose. But when diabetics often present with uncontrolled blood glucose levels, one of the common accompaniments is a low intravascular volume and hypotension. Now, diuretics, we talked about some of the medicines that can poison kidney transport proteins that exist within the luminal membrane of the nephron. So diuretics cause a loss of sodium in the urine, and we utilize that in clinical medicine to treat high blood pressure. Blood loss, well, that becomes obvious as you lose blood. About 50% of the total blood loss is composed of plasma volume, which is a sodium-rich fluid from the extracellular fluid compartment, and decreased intake. Of course, not as major an item, because indeed most of us can reduce our urinary sodium excretion to exquisitely low levels, but nevertheless something to think about. So how do you gain sodium? Well, in North America, we primarily gain sodium from our diet. Processed food, very high in sodium. This includes breads, cakes, biscuits, and fast food. And of course, many of us at different times in our lives have had too much fast food. The fries, the hamburgers, Chinese takeout, a lot of sodium content in these meals. And of course, the sodium is important for adding taste. And therefore, one of the things that captures us when we eat these foods is indeed the high sodium content. So let's begin to see if we can understand sodium balance and look at overall sodium balance by the kidney. Now here we return to the nephron, because indeed we remember this is where in the glomerulus, this is where the capillary network is going to produce the glomerular ultrafiltrate on the basis of Starling forces and put a filtrate into the lumen of the tubule so that it will wander through the nephron. Now, the main nephron segment, so the proximal tubule, is the workhorse where about 70% of the sodium that is presented to Bowman space and then begins to transit the nephron is reabsorbed within the proximal tubule. The loop of Henle collectively reabsorbs about 20 to 30%. The distal nephron, the distal convoluted tubule, about 5 to 10%. And then the collecting duct is really where the fine-tuning of sodium excretion occurs, where about 1 to 3% of the filtered load is reabsorbed. So we're going to talk about these nephron segments and try to understand how sodium handling moves about the set point. What is it that causes these nephron segments to increase sodium reabsorption so that there is relatively little sodium excreted at the end of the day, if indeed the proper signals are provided to the kidney. Now, imagine here's our nephron within its sort of normal anatomical configuration. Imagine if I grabbed the collecting duct and I grabbed Bowman space and I sort of pulled them out so that instead of having this convoluted proximal tubule, a loop of Henle, a convoluted distal tubule, and a collecting duct, I pull them out like an accordion. So this becomes Bowman space, and this becomes the collecting duct. And if we look at the filtered sodium on the y-axis, and we look at the distance along the nephron, what do we begin to see in terms of the sodium that is retained within the lumen of the nephron? It's almost as if you can imagine yourself sitting in some fantastic voyage where we're sitting there in Bowman space, surrounded by sodium. And as we transit that nephron towards the terminal segments of the nephron, how much sodium is retained. And so you can see that actually as we move through the proximal tubule, there's a tremendous amount of sodium reabsorption. As we get into the distal, the thick ascending limb and the loop of Henle, there's even more. We get into the distal convoluted tubule, the sodium continues to be reabsorbed until we're in the collecting duct, where we can see that we can reduce the remaining sodium relative to what was presented to Bowman space to a value as low as 1% or less. And when we look at the nephron, we begin to say, okay, well, how is sodium reclaimed from the lumen as water and salt move from Bowman space to the terminal segment, so the collecting duct. And here, stretched along, are the sodium transporters. So the key sodium transporters are sodium hydrogen exchangers, the NHE1 in the proximal tubule, that reabsorbs sodium down a concentration gradient and secretes hydrogen ion into the lumen of the nephron. We're going to come back to that in a lecture down the road. And then we have the sodium, in this case S is for sugar, but we have a whole series of cotransporters. Again, these cotransporters utilize the sodium gradient between the sodium concentration in the lumen and the low intracellular sodium concentration. And so this represents the luminal membrane, and of course on the basolateral membrane, that's where sodium potassium ATPase resides, which of course is responsible for maintaining the low intracellular sodium concentration. So as the tubular lumen sees the glomerular ultrafiltrate, there's a concentration gradient dependent sodium reabsorption, and that leads to the reclamation, if you like, of sodium. And as sodium is reabsorbed, the water follows along. The water follows both transcellularly and actually moves between the cells. Now, when we get to the thick ascending limb, how do we reclaim sodium? Well, that's another cotransporter, but a little bit more complicated than the sodium sugar transporters. Here we have sodium 2-chloride potassium transporter. Again, what drives the activity of this luminal transport protein? The low intracellular sodium concentration. Again, because all of these tubular segments have basolateral sodium potassium ATPase. And then as we enter the distal convoluted tubule, we see a number of other transporters, and the critical one that we're going to focus on here is this middle one, the sodium chloride cotransporter. It is responsible for reclaiming sodium down a concentration gradient in the distal convoluted tubule. Now remember, this transporter is inhibited by furosemide, and this transporter is inhibited by hydrochlorothiazide. And then as we move into the collecting duct, the key thing to remember is what we have are a sodium channel. In other words, not a transport protein that either is a cotransporter, an antiporter, but is rather a sodium channel. You can imagine if you have a cell sitting there with a low intracellular sodium concentration, and you punch a channel through that lipid bilayer that allows sodium to move down its concentration gradient, sodium will move across. And this turns out the biology of the sodium transport proteins, which is the epithelial sodium channel in the collecting duct, plays a very, very important role in fine-tuning the sodium concentration within the lumen of the kidney tubule. So, overall, Bowman space produces an ultrafiltrate, and then of course this is the final urine that is transported into the pelvis of the kidney, down the ureter and into the bladder. So 150 liters a day, I've put 140 millimoles of sodium in each of us, in each liter, and about 2100 millimoles. And that the set point for you and I is that less than 1% of that is excreted, but 1% is about 150 millimoles per liter, so that as the sodium intake is 150 millimoles per liter, ignoring other sources of sodium loss, the kidney excretes the same to balance sodium, so that day to day, you can imagine as we go along, the kidney, if you and I present our kidneys with 150 millimoles of sodium a day, then the kidneys excrete that 150 millimoles per day, and we stay within sodium balance. You can imagine that if we don't stay within sodium balance, if we eat this amount, excrete this amount, then our total sodium will increase in our bodies. So, here's the big question we have for ourselves. If the kidney has a set point in which it typically reabsorbs at least up to 99% or more of the sodium presented, how does it sense a sodium deficit? And then how does it respond to that sodium deficit by lowering the amount of sodium excreted? Said another way, how does it respond to even further increased sodium reabsorption by the kidney tubule? And that's what we're going to talk about today. So one of the ways I think about this is just imagine, this is a picture from the Australian outback, and let's just imagine you and I are on a long trek in which we're just going to walk down this road, and we're not going to have any water at hand, and we're not going to have any sodium at hand. Indeed, this is the kind of environment where you and I might imagine we evolved somewhat similarly, certainly an environment with relatively little water and relatively little sodium. Probably the explanation for why our kidneys are so good at reabsorbing salt and water. So here's the example we're going to try to address. Here's, of course, our nephron to remind us of the anatomy. Here's going to be our filtered volume. Here's our sodium concentration in each liter. Here's what we filter. And what we're going to do is say, well, what happens to, how do we increase the reabsorption of sodium so that instead of excreting 150 millimoles, we only excrete 10 millimoles. Why is that important as we're trekking across the desert? Well, we're sweating, we're losing sodium, and we want the kidney to receive a signal that tells us that instead of reabsorbing 99%, we reabsorb 99.95%, so that we only end up with about 10 milliosmoles. We conserve the sodium. You can imagine we would want a system that allows our kidneys in a relatively important way to reabsorb sodium, to reclaim all the sodium that is filtered for practical purposes, to avoid a progressive decrease in our vascular volume, a progressive decrease in our blood pressure, and a progressive decrease in organ perfusion. So how do our bodies sense a loss or excessive sodium and signal to the kidney to reabsorb sodium so that the excretion rate falls from 150 millimoles a day to 10 millimoles a day? So let's, at this point, return to our central concepts about body fluid compartments. And in this particular diagram, which I've showed you several times, we're going to look at the plasma volume. It turns out that the way in which you and I monitor our sodium balance is by gauging the plasma volume. That we have a particular physiology in place that allows us to determine whether that volume is high or whether that volume is low. And the reason that that's important is, of course, these are the sodium-containing body fluid compartments. So in order to sense a sodium deficit, we have physiologic responses in place that allow us to gauge the volume in the vascular or plasma compartment and then signal to the kidney. So how do we sense the volume in the extracellular space? And maybe even a little bit more specifically, how do we sense the volume in the plasma compartment or the vascular volume? So, well, where are the sensors? That's the first thing we would ask. And so you've talked a little bit in the cardiology physiology lectures. There are arterial baroreceptors. So you and I have baroreceptors that sit within the carotid body and within the aortic arch. So central arterial baroreceptors that respond depending upon how full they are. You can almost imagine, the analogy I use is of a balloon. Imagine that the carotid body is like a balloon, and it has an ability to respond depending on whether the balloon is small or whether it's big. So if the plasma volume increases, then the carotid body volume will increase in a sense, and you'll be able to sense that. So let's take a look at that. So in this particular, on the left-hand side here, we have two areas which are shown in yellow. These represent the carotid bodies here, the internal carotid, and these represent the aortic arch baroreceptors. And what they sense is pressure and volume. So you can imagine, as there's expansion in these areas, they increase nerve traffic to the brain, and we're going to talk about that a little bit more specifically. So they're sensing just how full the plasma compartment is in the arch of the aorta and the carotid body. And you can imagine actually why these, in evolution, why they might be there. This is a little bit about teleology, but it's interesting because, of course, the heart is here. So this is really registering how much pressure and what volume exists within the aorta, just as the cardiac output is generated. And up here, this becomes important because this is gauging the amount of fluid in the arterial structures that are delivering blood to the brain. So you and I want to know what our cardiac output is doing, and we absolutely want to know how much blood is being delivered and what is the plasma volume compartment in critical blood vessels that deliver blood to our brains. So pressure and volume. So let's look at this a little bit more. The first thing to note is that these baroreceptors communicate through the vagus nerve and through the 9th cranial nerve. So there is afferent arteriolar neurologic traffic from the baroreceptors to the brainstem, from the aortic arch through the vagus nerve to the brainstem, and from the carotid sinus receptors through the 9th cranial nerve to the brainstem. The bigger the balloon, if you like, the bigger the volume and pressure sensed within these receptors, the more the nerve traffic that is delivered via the vagus nerve and the carotid sinus, or the 9th cranial nerve, to the brainstem. The bigger the volume and pressure, the more the nerve traffic that is sent afferently to the brainstem. Now, what happens in the brainstem is that indeed there's an outflow tract that influences dominantly, and I want to focus on the sympathetic nervous system. So the cardioregulatory center within the medulla oblongata of the brainstem receives the nerve traffic via the vagus nerve and the 9th cranial nerve. And this nerve traffic from the arch of the aortic baroreceptors and the carotid bodies provides a negative influence. In other words, the greater the nerve traffic that reaches the brainstem from the carotid body and from the arch of the aorta, the more it delivers a negative influence or a downregulation of activity within the cardioregulatory center, and the more it reduces sympathetic nerve fiber output. So high volume and pressure, increase nerve traffic, a block, if you like, or a decrease in the sympathetic nervous system outflow from the cardioregulatory center of the medulla oblongata. So the consequence here of too much volume is to reduce sympathetic nervous system outflow. Now, let's do the opposite. If the volume and pressures decrease in the arch of the aorta and the carotid body, then the nerve traffic will be diminished. This nerve traffic through the vagus nerve and the 9th cranial nerve provides an inhibitory influence. So if you decrease an inhibitory influence, you increase sympathetic nervous outflow from the cardioregulatory center of the brainstem. So if we take that balloon at a set point, if that balloon shrinks, there'll be less inhibitory nerve traffic to the brainstem, and the sympathetic nervous system will be upregulated. If those carotid body and aortic arch baroreceptors sense more pressure and flow, there will be increased nerve traffic to the brainstem, and that will serve as a negative inhibitory influence and reduce sympathetic nervous system output. So, how does increased sympathetic nervous system activation influence how the kidney handles sodium? Again, decrease in carotid and aortic arch baroreceptor activity unleashes the sympathetic nervous system because it's telling us that that balloon is too empty. And what does it want to do? It wants to fill up that balloon towards the set point. The sympathetic nervous system has a critical role because it leads to the secretion of renin by the kidney. Renin is a proteolytic enzyme that is present within the afferent arteriole and actually within the juxtaglomerular apparatus, but predominantly in the afferent arteriole. Renin then is secreted and acts on a protein produced in the liver called angiotensinogen, and it produces a 10 amino acid peptide called angiotensin 1. And angiotensin 1 is rapidly cleaved to generate a 8 amino acid peptide called angiotensin 2. And angiotensin 2 has important functions that serve to increase salt and with it water reabsorption by the kidney. So when baroreceptors in the arch of the aorta are offloaded, the sympathetic nervous system is increased, and that leads to an increase in angiotensin 2, and that leads to an increase in sodium reabsorption, trying to return you and I to that set point, and that set point of sodium balance. So, where are these other afferent arterioles? Well, actually, there are other afferent arteriole or baroreceptors in the afferent arteriole of the kidney. So not only do you have baroreceptors sitting in the arch of the aorta, but you have baroreceptors sitting in the afferent arteriole. If the kidney doesn't see those pressures and flows, those cells in the afferent arteriole will increase their secretion of renin and serve to drive the generation of angiotensin 2, which when it reaches the kidney will serve to increase sodium reabsorption. This is an example that's shown right here. This is a light micrograph of an afferent arteriole, and this has been stained with an antibody for renin, and you can see the renin within these afferent arterioles. So if the pressures and flows fall in these afferent arterioles, those afferent arterioles release renin into the circulation, and that drives the generation of angiotensin 2. So low pressures and flows within the arterial circulation serve to converge on the kidney to increase activation of the renin-angiotensin system as well as activation upstream of the sympathetic nervous system. So again, we have this combination of baroreceptors in the arch of the aorta, the carotid body, and the afferent arteriole, which all converge on the kidney by virtue of an increase in angiotensin 2 generation. Now, kidney cells have angiotensin type 1 receptors, and these are G protein coupled receptors, and angiotensin 2 interacts with those receptors. It causes vascular smooth muscle vasoconstriction, and it has important effects on tubular function that influence sodium reabsorption. So let's look at angiotensin 2, the activation of the renin-angiotensin system, and how, as you and I are walking down that road in the outback of Australia, how we can predict that actually our sympathetic nervous system activity will rise. We can predict that the activation of the renin-angiotensin system will rise. We'll have more renin and more angiotensin 2. And what is it doing to our kidneys? Well, the question becomes, how does it affect sodium balance? So, well, the first thing to recognize is that, in fact, angiotensin 2 has important effects on sodium transporter activity. So it does what you'd want it to do. Its increased concentration reflects activation of the sympathetic nervous system and release of renin by the kidney, because the baroreceptors are telling the kidney, indeed, or telling the brain and the kidney, that the arterial system has low flow, low volume, and low pressure. And it makes sense. So what do? Well, in the proximal tubule, remember, this is the site of most of the sodium reabsorption. And much of that sodium reabsorption is dependent upon sodium hydrogen exchanger, NHE3. There are many isoforms of the sodium hydrogen exchanger. And there are also, of course, sodium solute cotransporters. But this antiporter exchanger dominates here in the proximal tubule. And angiotensin 2 serves to drive that activity. So one of the consequences, and this shows a proximal tubule cell. Here's the basolateral membrane with the ubiquitous sodium, potassium ATPase, maintaining a low intracellular sodium concentration. This will move down a concentration gradient. Hydrogen will be exchanged. Angiotensin II and actually norepinephrine both serve to increase the activity of that transporter. So you can imagine, again, now an proximal tubule cell bathed in angiotensin II and increased norepinephrine because the sympathetic nervous system is activated will rev up, if you like, sodium-hydrogen antiporter activity and move more sodium into the cell where it will then be summarily pumped out into the basolateral membrane and then reabsorbed in the peritubular capillary. What about the other main place that angiotensin II directly influences sodium reabsorption, and that's in the distal convoluted tubule? Remember, about 5–10% of the sodium filtered into Bowman space and that begins to transit through the nephron is reabsorbed in the distal convoluted tubule. And remember, too, that the major sodium transport protein that exists within the luminal membrane of the distal convoluted tubule is the sodium chloride cotransporter, NCC, sodium chloride cotransporter, and it sits here. Now, if you were designing the system, you'd say, jeepers, I think it's a pretty good idea to make sure that angiotensin II activates that sodium chloride cotransporter. It's not direct like that. There are some kinases and other proteins involved in that activity. But the consequence of increased activation of the renin-angiotensin system and the generation of angiotensin II because of sympathetic nervous system activity and because of low pressures and volumes in the afferent arteriole, increased Ang2 increases sodium and chloride reabsorption of the distal convoluted tubule, moving the set point so that there's even less sodium exiting the distal convoluted tubule than would be the normal set point for physiologic regulation. Again, we're pretty dependent on that ubiquitous sodium-potassium ATPase. It's critical for maintaining the low intracellular sodium concentration that drives this cotransport activity. And then there's a little bit more complicated mechanism by which angiotensin II influences sodium handling in the proximal tubule. And it does so indirectly, not by influencing the activity of a transport protein in the luminal membrane, but by causing changes in renal hemodynamic function, and in particular causing constriction of the efferent arteriole. And this is shown here in this rather complicated picture of the afferent arteriole, the glomerulocapillaries, the efferent arteriole, swinging around to the peritubular capillary network. A critical function of angiotensin II is to increase the resistance in the efferent arteriole. By increasing resistance in the efferent arteriole, you lower the pressure in the postglomerular capillaries and the lower the pressure here. Now, one of the other consequences is you increase the filtration fraction here so that the efferent constriction causes two things that are critical from a Starling forces perspective. It lowers pressure here, but it also increases the oncotic pressure. So when you come around to the efferent, when you swing around into the peritubular capillary and the proximal tubule in close juxtaposition, the fluid, there's a Starling forces that favor the movement of water from the lumen into the peritubular capillary. One, because the hydrostatic pressures are low in the proximal tubule and low in the peritubular capillary, but favor this movement, and the oncotic pressure tends to pull the water from the lumen into the peritubular capillaries. So, angiotensin II influences the afferent arteriolar resistance, increases the oncotic pressure in the peritubular capillary because it causes efferent vasoconstriction, and it lowers hydrostatic pressure in the peritubular capillary. The low hydrostatic pressure and the high oncotic pressure tend to move fluid on the basis of Starling forces from the lumen between the cells into the peritubular capillaries. And this is outlined in more detail in this slide, and I'll let you study that at home when you look at the different pressures, but this is just to emphasize that, again, in the presence of angiotensin II, Starling forces favor the reabsorption of sodium and water from the peritubular capillary into the peritubular capillary from the lumen of the nephron by lowering peritubular capillary pressure and raising peritubular oncotic pressure. The proximal tubule is very leaky, so that differences in Starling forces between the lumen and the peritubular capillary can favor water movement across that semi-permeable or leaky tubule, and as a consequence, the sodium is dragged along. So salt and water reabsorption is hemodynamically dependent upon the level of angiotensin II in the proximal tubule. So angiotensin II has another important effect. We've said, well, gosh, what it does is it serves to enhance Starling forces that favor water movement and sodium pulled along with it from the proximal tubule into the peritubular capillary. The sodium that remains within the lumen of the nephron is actively reabsorbed by the sodium-hydrogen exchanger 3. And in the distal convoluted tubule, there's activation of sodium chloride cotransport. Now, angiotensin II has another effect, and this effect is a hormone is in the adrenal cortex, and it leads to the synthesis and release of a steroid hormone by the called aldosterone. Now, aldosterone is produced by glomerulosa cells within the cortex of the adrenal gland. So a consequence of angiotensin II is to go to the adrenal glands and lead to the secretion of aldosterone. Now, this brings us to the collecting duct. Reabsorbs 1 to 3% of the filtered load of sodium. That's actually important because remember, you and I only excrete about 1%. So the collecting duct has the ability to really alter total sodium reabsorption in addition to what happens in these upstream segments and what happens in peritubular capillary Starling forces. What's the luminal transporter in the collecting duct? It's the epithelial sodium channel, called ENaC for short, epithelial sodium channel. And aldosterone has two critical effects in this part of the nephron. There are aldosterone receptors within the cell. Aldosterone increases the number and probability that the sodium channels are open. So a collecting duct bathed in aldosterone has more epithelial sodium channels in the luminal membrane, and the chances are that they're open. Consequence, therefore, and the consequence is that the more channels and more open channels favor the movement of sodium down a concentration gradient from the lumen of the kidney into the tubular cell and enhance sodium reabsorption. This series of slides is meant to illustrate that. So what we have here is an interstitial fluid. Here's the lumen of the distal nephron. Here's the cortical collecting duct cell. These are called principal cells. And here's the blood, interstitial compartment. So what happens? Well, aldosterone, represented by the small circles here, is a steroid hormone, so it diffuses across the lipid bilayer and binds to aldosterone receptors within the principal cells of the collecting duct. What's the consequence of that? Well, the aldosterone complex with the aldosterone receptor is a transcription factor that influences the expression of genes. One of those key genes is actually the epithelial sodium channel. So a consequence of increased aldosterone, increased transcription of the epithelial sodium channel, and increased insertion of the epithelial sodium channel into the luminal membrane of the principal cell favors the reabsorption of sodium down its concentration gradient. It has another effect too. It actually tends to enhance sodium-potassium ATPase activity in the basolateral membrane of the cell. And that's important because that means that as you enhance sodium entry, you have more sodium-potassium ATPase activity, and that allows the sodium to remain low within the cell to provide that constant concentration gradient favoring reabsorption down the epithelial sodium channel. So you can see that in this particular case, new channels that tend to be more open favor reabsorption, and more sodium-potassium ATPase moves the sodium. So as aldosterone goes this way, influences these two effects, sodium goes that way. And the sum effect is that you increase sodium reabsorption. You tend to shift the collecting duct sodium reabsorption such that it favors re-entry of sodium into the interstitial fluid and into the blood compartment. So let's put it all together. Long trek physiology, you're walking down the road, you're sweating, you're losing sodium. You begin to sense that in the baroreceptors that reside within the arch of the aorta, a critical place to sense cardiac output. The carotid body is a critical place to assess blood flow to the brain. And the afferent arterioles of the kidney, a critical place to sense blood flow to the kidney. If the balloon's empty in those sites, there's a decrease in nerve traffic in the aortic arch and the carotid body back to the brainstem that offloads the sympathetic nerve, that offloads a negative influence on sympathetic nervous system activity and leads to increased activation of the renin-angiotensin system. And of course, the renin is secreted in response to low pressures and flows in the afferent arteriole. A consequence of aldosterone, well, it affects the proximal tubule directly by influencing sodium-hydrogen exchange, but also changing Starling forces that favor reabsorption of salt and water in the proximal tubule. It drives the distal convoluted tubule to reabsorb more sodium by virtue of its effect on the sodium chloride cotransporter, and it stimulates the release of aldosterone from the adrenal gland that then travels to the kidney, influencing sodium reabsorption in the collecting duct by virtue of inserting more sodium channels, more open sodium channels, and actually increase sodium-potassium ATPase. The sum effect of these is that you reabsorb more of the filtered load of sodium. So already you start at 99% and you move towards 100%, never getting quite to 100%. So just before we begin to get to the end of our presentation, let's look at the opposite side of the coin. Most of the ways in which sodium is regulated by the kidney evolved in a setting in which it was critically important for the kidney to reabsorb as much sodium as possible by virtue of you and I evolving in an environment in which sodium was rare. We're not really challenged during our evolution by too much sodium. But that becomes a much more critical issue today in modern medicine. But let's look a little bit at what happens when there's too much sodium. So the example here we'll look at is, we'll ask sort of two questions, but I'm going to focus on the latter. The question is, what happens when you eat too much sodium? Or said another way, what happens when you sit in a hot tub? Now that seems a peculiar question, but let's come to where that might be. So here we are, we're all relaxing in a hot tub, or we're in a swimming pool. And what happens in that swimming pool? Well, one of the things that happens is that the water pressure serves to push on your skin surface and in fact transmits pressure to the interstitial space. What's the consequence of that? Well, the increased interstitial pressure in your skin helps to change Starling forces. And the increased interstitial pressure will drive water from your interstitial space into the vascular compartment. So you won't add sodium to your total body water and total body fluid compartments. What you'll do is you'll shift water and sodium from the interstitial space into the vascular compartment. And so if you and I were to sit in a hot tub, we would translocate about 1 liter of fluid, maybe a little bit more, from our interstitial space into our vascular compartment. And therefore, those baroreceptors sitting in the arch of the aorta, the carotid bodies, or the afferent arterioles in the kidney will suddenly, instead of decompressing them, so to speak, will suddenly increase in volume and pressure. So let's take that and look at sodium. So here we filter 100, the sodium concentration is 140 millimoles per liter, 21,000 millimoles increase presented to the nephron, and we reabsorb almost 99% of it. What happens when we suddenly have 300 milliosmoles that we want to excrete? So this is the example of eating 150 milliosmoles and then taking 1 liter of fluid from the interstitial space and putting it in the intravascular compartment. And in that liter, there would be about 150 millimoles of sodium. So how does the kidney handle that? Well, this first thing is that you'll shut down with increased baroreceptor activation. So remember, as the balloon fills, as the baroreceptors in the arch of the aorta and the carotid bodies expand, that will increase nerve traffic via the cranial nerve and by the vagus nerve and the 9th cranial nerve to the brainstem. And that is a negative influence. So you'll increase an inhibitory influence of brainstem activity, and you'll reduce sympathetic nervous system activation. So sympathetic nervous system nerve traffic will decrease, and that will reduce the release of renin from the kidney. And so you'll turn off not only sympathetic nervous system activity with norepinephrine and epinephrine, but you'll reduce activation of the renin-angiotensin system. And that will serve to increase, or said the other way, that will serve in the opposite direction to what we talked about previously, will serve to decrease sodium reabsorption by reducing Starling forces in the peritubular capillaries, by reducing sodium-hydrogen exchange activity in the proximal tubule, by reducing sodium chloride cotransport in the distal convoluted tubule, and by reducing aldosterone concentrations, reduce the number and opening of the sodium channels in the epithelium of the cortical collecting duct. The epithelial sodium channel activity will go down. And all of those changes will conspire together to reduce sodium reabsorption, so that when you increase your intake, or you shift that sodium into the interstitium, your kidneys begin to put out more sodium to try to restore the intravascular volumes and pressures towards normal. Now, there's another volume system that we look at, and these are called atrial stretch receptors. And I think these are really critically important, and they're very important and of interest, of course, here at the University of Toronto. It turns out that there's the arterial baroreceptors and in the afferent arteriole, the arch of the aorta, and the carotid body, but there also are atrial myocytes within the atrium that secrete a hormone in response to their stretch. And what's critical is that these were discovered in Canada about 1980. And what's of interest, of course, they were discovered here in this building in Toronto in the Department of Physiology. Dr. Sonnenberg was a senior nephrology researcher with a particular interest in the cortical collecting duct. And de Bold was a scientist at Queen's University who actually observed these secretory granules within atrial myocytes and wondered what they'd do. So Dr. de Bold saw these secretory granules, and Dr. Sonnenberg was an expert in the handling of sodium by the kidney with a focus on the collecting duct. So they got together. They ground up the atria and infused the atria into a rat, and the rat started to pee. And the rat developed a saline or sodium diuresis, so their sodium excretion increased. So that atrial myocytes, when they stretch, secrete a hormone that signals to the kidney to reduce sodium reabsorption. If you and I sit in a hot tub, our atria will in fact stretch. And when they stretch, they'll release atrial natriuretic peptide, which is the hormone that is in those atrial myocytes, and that will cause the kidney to excrete sodium and with it some water. One of the reasons, indeed, that you actually, when kids hop in a pool, they have an extraordinary urge to pee because the expansion of the atria leads to the release of a hormone that influences sodium handling in the kidney. So, an increase in vascular volume, eating sodium or hopping in a hot tub, going for a swim, stretches your atrial myocytes. They secrete they secrete atrial natriuretic peptide, a Toronto and Queen's University discovery. The atrial natriuretic peptide causes vasodilatation. It inhibits aldosterone release, and it inhibits sodium reabsorption in the collecting duct. It also goes to the brainstem where it decreases sympathetic inhibitory activity, and it decreases salt and water appetite. All things that would serve to restore sodium balance. So how does atrial natriuretic peptide affect the collecting duct? Well, it turns out that, that there are receptors for epithelial. So here's the atrial natriuretic peptide receptor. It actually resides within the luminal membrane of the collecting duct. It generates cyclic GMP, and that blocks sodium, epithelial sodium transport activity, and it also goes here and blocks sodium potassium ATPase activity. In a sense, the opposite of what aldosterone does, and it also independently inhibits aldosterone. The consequence is that the tubule cell in the collecting duct becomes less sodium avid. It doesn't reabsorb much sodium, and again, it's going to release that sodium into the urinary space and allow the kidney to excrete sodium, because it's receiving a signal that suggests the intravascular volume is expanded. So, atrial natriuretic peptide really focuses on here, and it's a low pressure volume receptor response. So how accurately does the kidney maintain the extracellular fluid volume? Well, here's an experiment. If people are on a constant sodium diet, then you suddenly increase their sodium intake, then sodium excretion doesn't excrete immediately. It takes a while after the sodium volume, extracellular fluid volume increases, and so it takes several days. So if you and I were to jump our sodium intake from 150 millimoles a day to 300, it would take a few days before we actually retain enough sodium to actually drive mechanisms that would serve to increase sodium excretion and prevent a progressive rise in our sodium content. You can imagine if you start here and start here, if you didn't have any means by which you could increase the sodium reabsorption, you would progressively increase your sodium content. What happens is that you stepwise increase your sodium content until finally the excretion rate goes up and it matches the intake on a day-to-day basis, so that after a few days you'll restore your steady state at the cost of a somewhat expanded extracellular fluid volume. So let's go back to our construct. Here's our body fluid compartments. We invert it. We focus on the plasma volume with the green arrow. And we look at sodium intake. And we want to match that to sodium output. The key point to remember is that we focus on measures of the plasma volume, which is, of course, one of the key compartments in the extracellular space. And by having that ability to sense whether that volume is low or high, there are a number of physiologic responses that serve to influence the handling of sodium by the kidney to either increase it in the hopes that in a reduced plasma volume, increased sodium reabsorption will tend to restore the plasma volume, or in the setting in which the plasma volume is increased, there will be a corresponding increase in sodium excretion by the kidney that will serve to return it to steady state. So, having said that about sodium, how indeed it is regulated in terms of excess or deficit, let's go back to the case of the day. So why is she so sick? Well, she had a severe depletion of her sodium as a consequence of loss of diarrheal fluid. Remember, there was probably about 100 millimoles of sodium each liter, and she was losing a liter a day. That would lead to a very low extracellular fluid volume. And she requires really immediate sodium and water replacement so that someone with cholera will die if there is no ability to restore their intravascular sodium and water, to replace their sodium and water that they've lost as a consequence of the diarrheal fluid. So indeed, she'll need isotonic saline likely, and it's possible that that volume of isotonic saline can be as much as 10% of her total body weight. So if she weighs 35, let's say she weighs 40 kilograms, she might need at least 4 liters of sodium chloride in an isotonic solution to restore her pressure. And as she continues to lose, she'll continue to need that. So salt and water resuscitation is the cornerstone of the acute management of cholera. In the absence of that, she'll rapidly deplete her plasma volume, her blood pressure will fall, her brain perfusion will decrease, organ perfusion will increase, and actually she'll pass away at the end of that if there's no intervention. Now, one of the things that people have looked at is, okay, well, if you don't have a ready supply of intravenous fluids, like we would have, for example, in our emergency department here at the Toronto General, for example, or Toronto Western, what would you give her? Well, here's a bunch of choices. You have water, juice. So juice is, of course, water with sugar. Cola is water with goodness knows what else is put into cola. Soup is a high sodium containing solution with water. So what would you tend to want to use here? Well, you really would ideally want to use an oral rehydration fluid, and it contains all of these. So it's more akin to a soup. And indeed, you want sodium chloride, glucose to provide calories, potassium we haven't talked about yet, but we will be to provide that, and sodium citrate to provide her with some bicarbonate, which we'll also be talking about in subsequent lectures. So that would be your choice. So summary. Signals to the kidney regulate sodium excretion primarily by cardiovascular volume and pressure receptors. They sense the circulating volume of the vasculature. Dietary sodium does have an impact on extracellular fluid volume. Eventually, in a healthy kidney, we will compensate, but the price will be an increase in extracellular fluid volume. And in some individuals, that increase in the plasma volume will contribute to high blood pressure. Sodium depletion and excess states are extremely common. Sodium depletion was the common state during our evolution. Excess states have become the common problem in our modern world. Angiotensin II directly stimulates sodium reabsorption in multiple sites along the nephron. Aldosterone stimulates cortical collecting duct sodium reabsorption, and it's a downstream consequence of increased angiotensin II. And the other side of the coin, atrial natriuretic peptide, derived from stretch receptor cells in the atrium, inhibits sodium reabsorption from the medullary collecting duct and serves to excrete an excess of sodium if indeed the atria are stretched. All right, so here's our next case, and this will bring us to water. So I'll look forward to joining you for a discussion of how the kidney regulates water balance. Thank you.